So do I start, or would you like me to start?
I'm still waiting to see.
Okay,
okay, we're good. They're going live now, so we know how to.
Sorry.
Hello, welcome to the Palaeontological Association 2021 Manchester Conference. My name is Victoria Edgerton, and I'll be chairing this session on Mesozoic life. We're going to start off our morning with our first speaker, Michael Benton from University of Bristol, on the fossil evidence suggests the first amniote displayed extended egg retention. Whenever you're ready.
Okay. Here we go. So my title is a bit long-winded, but in fact, the short version is which came first, the amniote or the egg? And in a nutshell, the answer is the amniote. The egg came much later. So the amniotic egg is a very famous innovation that the amniotes possess, which are broadly the reptiles, birds, and mammals. And it has a very particular structure that will be familiar to many. And it marks the beginning of a major radiation of reptiles that happened from the middle of the Carboniferous. And it's often used as a statement about why these groups of early reptiles through the Permian were so hugely successful, particularly relating to the switch from humid to arid climates. And this was encapsulated in a classic paper by Al Romer called The Origin of the Amniote Egg. And his argument, which has often been repeated, including by myself in all the textbooks pretty much, is these eggs were from the start mineralized, hard-shelled, rigid-shelled eggs, and they enabled the first reptiles to escape the limitations of other tetrapods in the Carboniferous by creating a kind of small private pond in which the embryo could develop. But there is an alternative view. And this is called extended embryo retention. Now, this is something we're familiar with in mammals. The majority of mammals, of course, retain the embryo inside. And you can see in this comparison of the chick and human embryo that there are shared features. And extended embryo retention is a term that is applied where the embryo develops largely inside the mother. And at various points, the egg can be laid, or indeed can be retained right through to term, which is what we call viviparity or live bearing. And in a classic paper, Michel Laurat plotted this on a phylogeny and actually noted the distribution of extended embryo retention. It occurs in coelacanths, bracketing the tetrapods as a whole. It can occur in lissamphibians, as well as in various other groups, including in squamates. So I'll review some of these. And the argument from Laurat and others was that Romer was wrong. It was nothing to do with a mineralized egg in Carboniferous. This came much, much later. It was all to do with retention of the embryo inside the mother with special membranes to prevent rejection, immunological rejection, as in mammals, and that is, in fact, the basal or primitive state for amniotes, and that the rigid-shelled mineralized egg came much later. So here are the two models in outline, the terrestrial model of Romer on the left, the EER model on the right. And if you go right down to the bottom, this is the origins in water. And on the left-hand side, we have mineralized eggs from the start, which are shown by the purple arrows, and viviparity as a form of extended embryo retention arising multiple times subsequently. Whereas in the EER model, the amphibian type egg laid in water, frog spawn, et cetera, becomes an EER egg that may be soft-shelled or may be retained within the mother for a fair amount of time during development. And in fact, the mineralized egg, shown by the purple arrows, only comes much later. So this is well known to anatomists of vertebrates and people who study development, but in fact, it's still questionable, or paleontologists are perhaps not so aware of it. Because of course we think in these terms of there are the eggs, we've got birds, turtles, mammals, they all seem to share the same kind of mineralized egg, and hence obviously that must have originated back in the Carboniferous, even though we don't have fossil eggs. Note that the oldest fossil eggs are about 195 million years ago. So the counter argument is that, first of all, the eggs of monotremes, the prototherian mammals, actually are not particularly mineralized. They're primarily parchment with limited calcite deposition. So we have to be careful in including mammals as evidence for that retrodiction. And the same is true of the majority of lizards and snakes. Most of them lay these parchment eggs. So if you're not sure what a parchment egg looks like, here's a couple of examples. And in fact, amongst that group of 10,000 species, only the geckos lay rigid-shelled eggs. A recent paper by Norell and colleagues in Nature summed up the evidence for dinosaurs. Yes, later dinosaurs definitely had, many of them, hard-shelled eggs. They're common fossils and whole nests are studied. But in fact, the earliest eggs from dinosaurs appear to be soft-shelled, as they argued. And in their simple phylogeny showing ancestral states, notice the soft egg dominates all the early origins back down in the Triassic. And the same is true of pterosaurs, the same is true of other archosauromorphs. So we think today of that large group of reptiles and birds, crocodiles, birds, and possibly turtles form a clade. They all seem to have hard calcite-shelled eggs, therefore surely their ancestor did too. Well, maybe not. The dinosaurs are in there, their ancestor didn't. A variety of other early forms from the Mesozoic didn't either. So here's a viviparous tanistrophiid from China that we described a few years ago. Choristodeers, also within the archosauromorphs, appear to show every kind of parity mode that you could imagine, including laying mineralized eggs. This one that Baoyu Zhang and we are describing appears to be in a soft-shelled egg, and others show viviparity. And viviparity evolved in all the aquatic marine reptile groups. And in fact, mineralized eggs are missing from the fossil record. Of course, absence of evidence is not evidence of absence, but nonetheless, you might think mineralized eggshells would preserve. This point, though, could be disproved tomorrow if somebody discovers a Carboniferous or Permian egg. But the important things to know, which paleontologists have maybe not kept up with, is that you can flip-flop either way. We tend to think, surely, if the hard egg goes first, vivipary comes second by suppression of the egg. But in fact, there are many, many examples, including this one, where species within a genus of many, many lizards, many, many snakes, show both states. And hence, if you have a vipary and vivipary flip-flopping between the species, clearly you can go either way in terms of transitions. And Blackburn, who studied this for his entire life and documented parity states across squamates, finds 115 egg to vivipary switches and 93 the other way. So this can happen, it can go both ways. And developmental data, for example, in this paper by Gao and colleagues, shows that extended embryo retention is a very easy thing to regulate in terms of the genomic switching. And this maybe helps to understand how you can flip-flop, because all that happens is that in the top species, the embryo is retained to stage 3 or 4, at which point the egg is laid. But remember, the eggshell is only put over the membranes containing the embryo and the yolk sac at the very last minute before it's laid. In the related species, which is viviparous, in blue, notice that it continues through stages 3 and 4 right through to stage 5 and 6, and the juvenile lizard is given birth live. So this flip can go both ways. I think you can see it would be easy to switch. So there's a great list of reasons, observations on both modern amniotes and on fossils that appear to show that vivipary, extended embryo retention, and soft-shelled eggs are probably primitive to the calcite-shelled egg, as shown here with the baby turtle peeping out. Now, the final part of the talk is what we did, is ancestral state reconstruction. We've already hinted at that. This is a well-known approach that's widely used, has been widely used for a long time. And it's been used already in exploring the parity states of squamates. They're the most interesting group. Notice the complete random occurrence, it seems, of vivipary shown in red. This is a phylogeny of all the modern squamates, 10,000 of them. and vivipary is popping up all over the place. According to the first analysis by Pyron and Burbrink, they found vivipary was the basal state. Notice the red going down to the bottom. Whereas in a repeat a year later, King and Lee, using the same evidence, the same data, but slightly tweaking the analytical approach, found that ovipary was the primitive state. So we can have both. You can flip-flop between the two. But it depends on the methods. We have to be careful about the methods we use. So we did an analysis here of all amniotes, and you can see at the top, the mammals down through the marine reptiles, lizards, and in the middle, turtles and archosaurs towards the bottom with birds. And we ran multiple techniques to try and avoid the problems that had been seen in the squamate studies. And in this case, we used parsimony methods, maximum likelihood methods, and Bayesian methods, and we found fairly consistent results. If you look at the turquoise blue color, that represents the presence of extended embryo retention as seen in mammals and various other groups. And the pink color, which is vivipary, notice that they go right to the base in most of the analyses. So variable results, but we ran this multiple times and we got pretty much 90, 10, 80 to 100% likelihood of vivipary and extended embryo retention right to the base. The only extensions are amongst the birds at the bottom of the diagram and including the dinosaurs, where primitively they show the opposite of the pale pink for absence of extended embryo retention. We had some problems to resolve here, and thanks to Joe Keating and Armin Elsler, they worked out some of how to deal with it. We recorded extended embryo retention simply as a presence and absence, but we linked reproduction mode and eggshell mineralization. And the fact there is you can't have all possible combinations if you look at the list of states. Because if you have vivipary, you cannot have any kind of mineralization of an eggshell because there is no eggshell. So the technique that Joe developed based on previous publications was to allow us to run this multiple ways where all of these states are independent. Notice the pink boxes for vivipary and the other states are in the oviparous condition. And the switch on, so independence on the left, the switch on is where you can only have these states expressed as notional states in vivipary when they're present in oviparous forms.
And Mike, if you don't mind finishing.
Yep, I'm going to wrap up. Thank you. And so we find then that basically, having run this multiple times, this is the result. And in summary, here is the classic view, hard-shelled eggs everywhere. And the current view then, vivipary to the base, soft-shelled eggs to the base. Thank you.
Thank you, Mike. And just to remind all of our audience, when it comes to questions, if you can please log on to Slack, you'll have that information in your PalAss information packet. And if you go into Slack and if you scroll down to the channels and go to Mesozoic Life, you can ask people in live view. And so that you can see what other people are asking, and people can also then respond to your questions. And at the very end of the session, we will then have a group question and answer period. So thank you again, Mike, for such a great talk. And next we have Stefan Speakman. New information on the archosaurs from the late Triassic fissure fills locality of Pantophenum, Wales.
Yes, thank you very much for the introduction, Victoria. Let me just see if I can maximize this. Okay, here we go. Yes, so the location I want to talk to you about today, this general area is located in the Bristol Channel area, and there are a number of fissure fill sites from this region that date between the late Triassic and early Jurassic. As you can see, some of these fissure fill localities are located in the English side of the channel area, and some of them are located in the Welsh, the southern Welsh part of the Bristol Channel. And as you can also see is that during this time period, this region was likely an archipelago with the different sites being distributed on different islands in this region. And because these are fissure fills, they have quite a unique preservation where small-bodied animals or remains, including animal remains, were washed into caverns in Carboniferous limestone, and this is how they became preserved. So quite uniquely, it preserves in particular small-bodied terrestrial vertebrates. And these include some very famous forms such as Morganucodon, one of the earliest mammals, several rhynchocephalians, also one of the earliest named dinosaurs, Thecanthrosaurus, and Terrestrosukis, which is one of the earliest stem crocodilians, which is one of the taxa I'll be talking more about today. And because all these animals are all quite small-bodied, and we don't have any evidence of larger-bodied animals, including even isolated remains of larger-bodied animals, which one might expect would wash into these fissures. Some people have suggested that insular dwarfism might have played a role in the fauna on these islands, which has resulted in all these taxa being quite small compared to their mainland relatives. And the one site I want to talk about more specifically today is the late Triassic site at Pentefinnen in southern Wales, which is late Triassic in age, Rhaetian, although some authors suggest possibly even late Norian in age. And this fauna is dominated by a rhynchocephalian called Cleidosaurus. But in addition to this, one of the interesting aspects of this site is that it preserves the most diverse archosaur fauna from the fissiophils. So this includes Terrestrosukis, this crocodilomorph I was talking about, which is actually quite abundant at Pentefinnen. It also includes Enigmaspina, which is a small-bodied pseudosuchian that I will also be talking a bit about today. Then there's Pentydraco, which is a sauropodomorph dinosaur that was either very closely related to Thecanthrosaurus or might actually represent a juvenile form of Thecanthrosaurus. And then there's also a small theropod dinosaur that we recently described that I will also be talking a bit about today, which we named Pendrick milnerae. And I'll actually start out with the theropod. So here is the holotype of Pendrick. It consists, as you can see, of a very nicely preserved pelvis in left lateral view. In right lateral view, you can see that the pelvis is mostly broken away, which allows us to see the sacral vertebrae and the posterior dorsal vertebrae quite nicely. And then we also have this left femur that comes with this specimen. It was associated with the pelvis, and it fits nicely with the left side of the pelvis and the part of the same individual. And we also have two referred specimens with this exquisitely preserved dorsal vertebra on the left, and then a small fragment of an ischium on the right-hand side. So, of course, we performed a phylogenetic analysis in which we recovered Pendrick as a coelophysoid theropod, specifically a non-coelophysid coelophysoid theropod, so at the base of the Coelophysidae group. And in order to look more into this idea of potential island dwarfism in the fissiophils, we did a body size evolution. We looked into this using an ancestral state reconstruction analysis, using the femur length as a proxy for size. And from this analysis, we found that Pendrick is autapomorphically small. It is a very small-bodied taxon, and as you can see, its closest node was considerably larger. But Pendrick is not unique in this regard. There are several coelophysoids that show the same. There's Povovenator as well. There's Pocomsignathus as well. And in addition to this, it's also unclear exactly how old the holotype of Pendrick was when it died. We know it wasn't a very early juvenile. We also know it wasn't likely fully mature. So these are all factors that kind of complicate this analysis. So in that regard, the final conclusion was that these results are ambiguous when it comes to evidence for insular dwarfism. We don't quite know whether island dwarfism was occurring on these islands based on this analysis alone. So we need more research into this. And here is a reconstruction of Pendrick in its natural environment. Now we're moving on to Enigmaspina, which was actually recently described by Patrick et al. In 2019. And in it was described as an enigmatic pseudosuchian archosaur. However, they couldn't really pin down the phylogenetic placement of this taxon. Depending on the analysis used, it was either found among Proterosampsians or ornithosuchids. And one of the interesting aspects of this taxon, which actually might suggest an even different phylogenetic placement, is the presence of these bifurcated neural spines on the cervical vertebrae and the anterior dorsal vertebrae, which is actually a trait we mainly see in Herpetosuchids. So these were all reasons why we thought it was wise to reconsider Enigmaspina and maybe say whether we can say a bit more about where this taxon might belong and what else there is to say about this taxon. So we took the data, we were able to get the data from Patrick et al., the CT data that they produced, and we used it to reconstruct the skeleton in a bit more detail. So here we have the reconstructed vertebral column with the ribs and the osteoderms and the scapula. And we looked a bit more specifically at these osteoderms, which are quite peculiar and quite unique among early archosaurs, in that they have that they're bifurcating posteriorly at the back. And if you look at them, you can see this curvature at the anterior end of the osteoderms, which seems to indicate that they would have overlapped like so in a single row, which is also quite unique among archosaurs as far as we can tell. In our phylogenetic revision, and this is an ongoing project, we have recovered enigmaspina at the very base of one of the clades within Pseudotsuchia, and this is the clade consisting of Ornithosuchids, Herpetosuchids, and Aetiformes, which is actually quite interesting because now we know which part of the Pseudotsuchian tree it belongs to, and also it seems to indicate that it doesn't belong to any of these clades specifically, because it has features that somewhat differ from all these groups, indicating that it might actually be something new for this part of the tree, which is quite interesting. And then we move to Terrestrosuchus, which is the bulk of my project at the moment. As I said, Terrestrosuchus is quite well known from Pentaphynon. We have on the right-hand side here, we have the holotype, and you can see that those are disarticulated but somewhat associated skeletons, including skull elements as well as postcrania. We have another nice block here showing, among other things, a very nice scapula. And then we also have some isolated remains, such as this very, very nice but extremely fragile little humerus here. And here in the top left, you see reconstruction of Terrestrosuchus. So you can see this was a gracile, terrestrial, probably quite an active, small-bodied crocodilomorph. And here we have a reconstruction of it in Pentaphynon on the top right. So one of the things we're doing is we're looking more into Terrestrosuchus using CT scanning. So we have this very nice specimen on the top left, and particularly the skull seemed very promising. So we decided to CT scan it. The scan was quite hard to work on, so it took me a lot of time to work it out in the end, but I think the results paid off, because as you can see, we have the skull quite nicely here in lateral view, and here in medial view, and even we have somewhat of an onset of reconstructing the skull based on the material in this one scan. And you can see that the backside of the skull is essentially complete, the temporal region and the orbital region. So here are some of the more interesting findings that we have so far. Based on this scan, we have very nice quadrates, as you can see, and we can indicate that the quadrate was located quite medially in the skull, as we can see based on this nicely preserved quadrate facet on the medial side of the right squamosal. And this is actually a trait that is quite typical of crocodilomorphs. And in more derived crocodiles, we see that the quadrate actually fuses to the brain case. And this has not yet happened in Terrestrosuchus. So that is quite interesting in that regard, that it's somewhat intermediate in that respect. Furthermore, the skull is quite heavily pneumatized, as you can see from the basis sphenoid here, as well as this cavity within this opening, the foramen in the lateral sphenoid as well.
1 minute.
Yeah. And now this is basically what we've done so far. This is an ongoing project. And what we want to do now is we want to finish this revision of Terrestrosuchus as well as enigmaspina from Pentaphynon. And then once we've done this, we want to continue and look at more of the archosaurs from the other fissiophils. For instance, there's more crocodilomorph material from Cromhole, which likely represents a new taxon. This was already suggested in the past, but also some initial analysis seem to indicate this. If you look at the carpus, for instance, the wrist, we can see that the radiali and ulnari are quite different in Terrestrosuchus relative to the Cromhole crocodilomorph. So this is quite interesting. We also want to do more histology on these crocodilomorphs in particular, because we have quite abundant material, and it would be so interesting to say something about the life history of these animals. And then finally, further down the line, we also want to tackle the island rule in more detail by looking at the entire fauna rather than just at individual taxons. And with that, I would like to thank my funding body as well as my collaborators. And thank you very much for listening to me on that Sunday morning.
Thank you, Stefan. Again, just to remind all of our viewers that you can all go and ask questions in the Slack channel for under Talks 02 Mesozoic Life. And all of our speakers will be there answering your questions. And we'll also have a question and answer period from 10:12 to 10:30. So if you have any questions, you can save those questions for then as well. So now moving on, let's welcome Thomas Halliday for Dinosaur Entomology Reflects and Reinforces Global Inequalities in Paleontology. Take it away, Thomas.
Okay, hello everybody. Thank you for coming to listen to this talk. On something a little bit different maybe from the rest of the fair in this Mesozoic session, but which I think is very important from a perspective as us as scientists in local and global communities, from a social perspective. So we're all familiar really with scientific nomenclature, the binomial system that gives all of the specimens we study a name. And these names really have two functions, right? The primary function really is as a label for a clade. So we're talking about, for example, this trilobite, then we would say that this is Elrathina athena. It has a name which essentially refers to that holotype, and everything else that we give that name is similar enough to that holotype to deserve that name. But it also has a secondary meaning. It's an aid memoir for scientists. Now, these names are governed by the ICZN, and the ICZN has a series of articles, which are essentially the rules that you have to follow in order for the name to be valid, but it also has recommendations, which are things that we ought to follow. One of these is responsibility of authors informing new names. And so I want to draw your attention to this. So it says that authors should exercise reasonable care and consideration. We should, informing new names, we should consider the people that are going to be using that name after us, and that assume and to produce names that are appropriate to the fossil, compact, they're not really long, that they sound nice, they're memorable, and they do not cause offense. And this last one is, I think, particularly important when we're considering the history of colonialism in paleontology, which I will come on to in a moment. So when we're coming up with a name, that name that we give for our own memory, it can have a series of possible meanings. We can decide to say something about the fossil that is factual, something about its observed anatomy, something about its age or where it comes from. We can give it something to do with a hypothesis. For example, we can hypothesize a relationship with another taxon. We can hypothesize an ecological trait, so its diet or its locomotion, or we can say something about the environment that it's hypothesized to have lived in. And then the third category really is that we can honor something or anything, really. I mean, all of those pictures there on the right-hand side are things that have been honored in the names of dinosaurs, whether we're talking about Egyptian gods or paleontologists or companies or cultures. Now, honoring, is an active choice. And we really want to know here who gets honored and who does the honoring. In a recent study, 2020, there's a DOI link there for anyone who wants to screenshot that and look at it later. Looking at modern dinosaurs, birds, found that within the last 30 years, 96% of those species that honor someone are species from the global south. However, 62% of those are authors from the global north honoring people from the global north. So we have individuals coming in from other countries, studying the species there and naming them after species from their own countries or from other northern countries. And when we talk about scientific colonialism, what we're really talking about is a disconnect between the place itself and where the center of knowledge for that place is, which has all sorts of social implications that are obviously very important. And paleontology has quite a long history of this kind of geographic disconnect. So dating back to the 18th century, that's the Comte de Buffon, who was a big promoter of the idea that the Americas just produced degenerate animals, nothing as big as you could see in the old world. And he extended that idea to people. Moving on into later, the sort of the boom years of early paleontological research, so you have, of course, things like the Tendaguru expedition, which was the largest dinosaur excavation to ever take place in what is now Tanzania and what was then German East Africa. And it was most of the specimens from there, indeed, I think pretty much all of the specimens from there are now held in Germany, such as iconic species like Brachiosaurus, or genera like Brachiosaurus, Giraffatitan, as it now is. And of course, recently, this is not something which has stopped. Very recently, it hit the headlines with Ubirajara being a specimen that was illegally smuggled out of Brazil. And in the end, the paper that described that specimen was withdrawn, and that has further implications for whether the name can even be considered valid in the future as well. So this has real practical importance as well as ethical importance within our subject. There's also various more abstract neo-colonial activities in paleontology. So we can talk about parachute science, which is where you have researchers that are based in one country going and conducting research elsewhere, or indeed the sort of the impact of where the fossils are coming from. A lot of fossils come from mines, and ExxonMobil at the moment owns a major area which is being excavated for dinosaur fossils. Their presence there, their fracking there is really damaging local communities. It is against the wishes of all of the people in that local community. And yet there is a dinosaur which is named after ExxonMobil from that site in honor of their support of paleontology. Now, why would I look at dinosaurs particularly for this? Well, dinosaurs hit the headlines every time a dinosaur is named. These are a few examples from only one newspaper in the UK from the last few months, since October, of reports of new dinosaurs being named. And of course, they're some of the few extinct creatures where the name is actually known by people at large. So what we did as a group to try and investigate what the effect of this colonial history is on etymology and naming practices is we have created a comprehensive data set of all naming events within Mesozoic dinosaurs, so including birds. And we do also have data for eggs and traces, but we are just restricting ourselves to body fossils for this purposes. So for example, these here are two dinosaur species that we have where we've recorded locality, we've located where it currently is, the etymology of both the genus and the species, and the affiliations of the authors, and the reason for honoring people or things when they are honored. And I just want to say that we are looking at direct etymology. So for example, this is Vegavisiae, which is a bird from Antarctica. It is named after Vega Island, where it was found. But of course, if you wanted to be really specific, you'd say Vega Island is named after a Swedish ship, the Vega, which had polar exploration history, and that of course is named after the star Vega, which is named after the fact that it looks a little bit like a flying vulture when you look at the stars around it. But we're just concentrating on the very specific local, that would be a geographic name. Vegavis is named after the island. So what can we see with trends over time? Well, first of all, we can say that the number of species, the number of genera that are being honored has increased slightly over time. There's a slight step change that occurs from the 70s onwards. And in fact, if you do look at those on the right, you can see that when you look at the absolute numbers, because we are naming far more dinosaurs now than we ever have before, the number of eponyms, the number of honoring names is really going up. So this is something we do need to think about. Who are we honoring and why? Genera tend to be named more after location. They tend to be named they tend to be named more with folkloric eponyms and relationships. For example, Parasaurolophus is a relative of Saurolophus, whereas species tend to be the ones that are accruing these eponyms after real people. We can see trends in this data set of an increase in languages other than the traditional Latin or Greek names, and this is something which has gained pace in the last few years. And we can also ask interesting questions about trends within each country. But what I really want to look at for the purposes of this talk is here. So the orange lines on the graphs on the left are refer to countries that have benefited from imperialism, benefited from colonialism. The blue lines are countries that have not. And these are lines that show the proportion of papers, of descriptions in those decades which have an author from those countries, either first author, any author, or all author. So what we can see is, firstly, we have increased international collaboration through time, as reflected by the decrease in all authors coming from the same country, but also an increase in the any author from the previously colonized countries. However, it's still a lag. It's still below where we are for countries like the United States, like the United Kingdom. And if we look at specific individual countries, a lot of the old powerhouses of paleontology have this, like the UK, USA, UK, and Germany have this kind of pattern where any and all authors, this is for first author or any author, these two lines have a consistently high, it's almost everyone. There's almost no dinosaurs that are described from the US that do not have a US author. And countries like China and Argentina, the sort of emerging powers that have managed to consolidate their position in the paleontological world, have a brief period of where you get people from other countries naming it, and later on increase. But then there's countries like Mongolia and Uzbekistan, which are really suffering. I mean, Mongolia currently has no papers being described with first author from all of the dinosaurs that are described from there come from other countries. We also have we can look at names, for example. I'm going to skip over this because I'm slightly running out of time because I want to come on to a couple of case studies. We have examples in names of direct colonial extraction. Dicalosaurus lettofobecki is named after a soldier with a very, very shady imperial history. Majungasaurus crenatissimus was discovered during a literal invasion by the army field doctor and is named after the French name for the city, not the Malagasy name. We have political dinosaurs. Nipponosaurus sakhalinensis is a dinosaur whose name is laying claim to the southern half of the island of Sakhalin, which is disputed between Russia and Japan. We have dinosaurs that are named after the liberty, the bicentenaries of the independence of these countries from their imperial hubs in Europe. But what's important here is also perspective. So in 2022, dinosaurs were named Navajoceratops and Dinobolata. They are named after the same group of people. The Navajo Nation is an indigenous group in the United States who in 2017 voted not to change their official name to the Diné Nation. They call themselves Diné. Navajo is a sort of Hispanicized version of what other groups have called them. And indeed, of the 30 names that we can identify as being after a people, a culture, 4 are unambiguously offensive names within that culture. And this is problematic. These are not names that can be easily changed by the ICZN rules, and yet they're lingering. And this is not an old issue. It's not old people being racist. The oldest of those was named in 1993, and the others were named in the 2000s. So this is something which we really do need to consider deeply when we are looking at naming.
Thank you. Unfortunately, we have run out of time. I do apologize for that. It is very interesting.
Yep, I have so many examples that I can talk about in the questions.
Yes, exactly. I was going to say you can continue this amazing conversation in the questions section of Slack. Again, for those of you who are just joining us, you can go and refer back to and look at the questions and talk to the speakers in the Slack channel on the PalAS conference Slack channel. So you would look for the channel called Talks 02 Mesozoic Life. Thank you again so much, Thomas. So now moving on to going to next is Chinkoda, talking about simple and branched pterosaur feathers with distinct melanosome geometries reveal deep origins of visual signaling in feather evolution.
Thank you. Hi, everyone. Today I'm going to talk about something very exciting about a new pterosaur specimen and what it means for something quite controversial, the origin of feathers. So we all know what feathers are. We are familiar with feathers. They come in different shapes and different sizes. Feathers evolve from a follicle in the dermis. Feathers come also, they have very different functions. Some are shown here. But maybe the most interesting feature about their biology is how they evolved. So here I'm showing you maybe the most known model of feather evolution and development. This model shows how feathers evolved from rather simple morphologies like this monofilament to more complex morphologies. So if I go through the different stages, stage 1 is monofilament, stage 2 is a tuft of filament, then we have stage 3A and stage 3B. Stage 3A corresponds to a central shaft, a rachis with barbs, and stage 3B corresponds to barbs and barbules with no rachis. Then we have stage 4, where the barbules interlock to form a closed vein. And here the feather is symmetric. And then finally, the most complex stage is stage 5, where the feather is asymmetric. So that's a feature that's very important for flight in modern birds. So now, how are those different modern birds, modern feather morphologies traced in the fossil record? So here you have a phylogenetic tree for Avimetatarsalia, so the clade including all dinosaurs and pterosaurs. So here we just need to look at the colors. They each correspond to a different feather type. And if we look at here, the birds and late diverging theropods, you can see that they have quite complex morphologies, mostly closed vein feathers. Now, if we look at early diverging theropods, we can see that they had quite simple feather morphologies. And actually, there were also some intermediate morphologies that are not reported here in that Evo-Devo model. And maybe what is very intriguing maybe is that some pterosaurs show branched filaments. And the question is, were those filaments feathers or not? So here are some examples of feathers in the fossil record. Feathers are integumentary filaments, and all those integumentary structures contain melanosomes. And those melanosomes have different shapes, different geometries. So here, if I take as an example that unurinated pterosaur from the Middle Age Jurassic of China, those branched filaments were interpreted as feathers because they had melanosomes, but actually we are not. We still don't know for sure that those structures were feathers. And if they were feathers, what were they used for? Thermoregulation was one of the hypotheses that was suggested, but we still don't know if those filaments had other functions. So in our study, we wanted to look at two questions. What are those branch structures in pterosaurs, and what were they used for? So until now, we couldn't reply to those questions because we didn't have specimen to do so until now. So here we report a new specimen of Papejarid pterosaur. Those pterosaurs were quite weird. So they had, some of them had very large head crests, and that's what we have preserved here. That's what we were mostly interested in. So before I describe that specimen, I have to talk a little bit about its history. So that specimen was exported illegally from Brazil and then acquired by a fossil collector. That fossil collector then contacted the Museum of Natural Sciences in Brussels, in Belgium, and donated the specimen. And therefore, the museum contacted the Brazilian authorities and started the negotiation to repatriate the specimen. So this specimen has a collection number from a museum in Brazil and will be repatriated in early January, so in a few weeks. So this is the fossil we were looking at. So this is the posterior part of the cranium with the occipital process preserved here. We have as well part of the bony crest preserved. And we have a very large soft tissue crest extending here from the occipital process to the post-premaxillary process here. And this is the most complete known for this species. So here is a close-up of the region around the occipital process. So that's really the region that we were most interested in because it shows filaments. Oops, I think I have an issue with my PowerPoint. Can you see the slides? Maybe I should stop sharing and then I'll try to restart PowerPoint.
Yeah, if it's locked, go ahead and go for that.
Sorry about that.
Well, you get to show everyone your amazing backdrop.
Okay, so I'm back to the first slide. Okay, I will just go through all the slides I showed already. So I was here, I think, yeah. Okay, so we had, so we observed monofilaments here, but what was very interesting as well is that we observed not only monofilaments, but filaments that are clearly branched. So you can see here a close-up of those branched filaments. And so you can see that they are curved, so they seem flexible. They are splayed out at the side here. And you can see that the shaft here is quite thin. And so this splaying structure really is characteristic of branching along the length. Here is another example of a branched structure in that pterosaur. So you can see the branches here along the shaft. So that's branching along the length really is characteristic of feathers. So here we can answer to our first question: are those structures feathers? Yes. Branching along the length confirms that those structures are feathers. So we compare those filaments, those feathers, to the Evo-Devo model of Crumb. The monofilaments correspond to stage 1, and the branch feathers to stage 3A, so an open vein feather without barbules. So here is our full reconstruction. So again, we just need to look at the node here and the pterosaur here. And what we observed is that the most likely result of our reconstruction is for the ancestor of pterosaurs to have monofilaments, brush-like filaments, and tufts or tufts of filaments. So branching is really an ancestral feature of feathers.
1 minute.
So then we looked at those structures and we observed melanosomes in each of them. The melanosomes have different shapes. So they are ovoid in the skin, they are elongate in the monofilaments, and they are ovoid again in the branched feathers. Here we have another ancestral stage reconstruction, but for the diversity of melanosome here. And you can see that the most likely result is to have a diverse geometry for melanosomes. So what we know about melanosomes in pterosaurs is that those unurinated pterosaurs had one melanosome geometry, or pterosaur has two melanosome geometries. So it seems that there was an increase in diversity through time. We have two types of feathers here. We have two types of melanosome geometries. So it seems that the ability really for feathers to vary melanosome shape was already present in pterosaurs and probably in their common ancestor to pterosaurs and dinosaurs. So the ability of feathers to vary melanosome shape and then color is an ancestral feature. So this really suggests that the ancestral functions of feathers were the production of color and the variation of color on the body. So to conclude, if we go back to our questions, are the branched structures in terms of feathers? Yes. And what were they used for? They were used for production of color and variation of color. So finally, with our study, we can say that a diverse melanosome geometry in feathers is ancestral. The genes involved in melanin-based coloration were already in place very early in the ancestor of pterosaurs and dinosaurs. And then that visual display was likely a very important function of feathers in pterosaurs.
Thank you, Oud. That was fantastic and really interesting. What an amazing specimen that you have. And I just want to remind our audience that if you have any questions for Oud or any of our other incredible speakers, you can go on to the Slack website and go to follow the links to go down to the channel of Talks 02 Mesozoic Life. We've had some great discussions going on in the chat that I've been following. So please ask Oud some incredible questions about this amazing specimen. So moving on to our next speaker. So our next speaker is Titiwut Sedtapan-Nitsan-kun, also known as Perth. And he will be talking about a juvenile rhynchosaur from the Otter Sandstone of Southern England. Thank you, and take it away, Perth.
Thank you very much. Yes, so I will be presenting on a juvenile rhynchosaur specimen from the Otter Sandstones of Southern England. There we go. So I'd first like to quickly cover some important factors about rhynchosaurs. So the end Permian extinction was a devastating mass extinction that took out roughly 70% of all terrestrial life on Earth. And the recovery process changes the ecosystem drastically, in the sense that there are now a few major players within the ecosystem itself that prevent further collapse as the diversity within the ecosystems continue to improve. And one of those players was rhynchosaurs. Now, rhynchosaurs themselves are basal archosauromorphs that have curved jaws, 2 to 3 rows of unique dentition, beak. They have digging toes in their hind limbs, and they have actually been found widespread across the globe. And in many cases, they've also been found living in large herds. As you can see, rhynchosaurs are limited to the Triassic period, diversifying rapidly during the Middle Triassic before going extinct in the Late Triassic. And one of the things that kind of led to their success is their dentition. Rhynchosaurs have what is known as ankylosthecodont dentition, meaning the teeth are deeply rooted and they're worn down to form a shearing plane. The tooth replacement itself occurs in batteries like this. And as the jaw continues to curve forward and grow longer, the teeth shift forward and new teeth come in from the back. And tooth replacement itself only occurs in the posterior end of the jaw. And here's another graphic here that shows us the sort of shearing plane. Both the tooth and the bone surrounding the tooth is actually worn down. The Otter Sandstones where this specimen was found is Middle Triassic in age. It's part of the Helsby Sandstone Formation, and the specimen was found and donated by Rob Coram. The specimen was found, in the High Peak section at beach level in sediment representative of detrital channel lag, most likely from a flash flood. The otter sandstones are known for two types of Rhynchosaurus, Phodonyx and Bentonyx. And as you see, we will be using Bentonyx skull, mostly because the skull, this is one of the more complete Rhynchosaurus skulls from the otter sandstones. And although there are differences between Bentonyx and Phodonyx, we deem them to be more ontogenetic, most likely not showing up in the juvenile itself. Onto the specimen in question. Here it is, as you can see by the scale bar, it is a tiny specimen, only about 6 centimeters in length. It is very fragile, and because of that, we decided that it would be best to CT scan the specimen and segment out the bones instead. So after about a year of work, here we have it. As you can see, the specimen itself is relatively complete. We're lacking a lot of the limb bones and a lot of the digits, but the pectoral girdle is very well preserved. The sacral area is much more distorted, but still well preserved, and the skull is exceptionally preserved. One of the big questions we had to answer was, is it actually a rhynchosaur? But using cladistical analysis from Escora in 2016, we find that features and characteristics of the maxilla, ilium, jugal, and the scapula indicate that it most likely belonged in Rhynchosauria. Now, the skull is very, very well preserved. We're missing the frontal beak. That would be very Rhynchosaurian. We're missing the palate and the brain case. That wasn't very well preserved. And now when we put the skull side by side with an adult, you can see a lot more differences, which kind of explains why we had to double check if it is actually a Rhynchosaur. One of the big things that you'd notice right away is the teeth. The teeth are large, they're pointy, whereas in the adult, even with the unworn teeth, the teeth are more rounded with an enamel cap. The teeth also are located throughout the maxilla, whereas in the adult, it's actually located in the mid and posterior section only, and towards the front, the teeth actually are much more worn down, and either that or reabsorbed. Looking deeper into the skull, we find that even more differences. First off, the juvenile dentition does not show any replacement teeth like we see in the adult. And the teeth actually appear more acrodont instead of ankylethecodont. So the teeth actually kind of just sits on the dentary itself. The juvenile exhibits no tooth wear, and the teeth are actually kind of more hollow. This suggests that the juvenile may be even younger than we realize, possibly even a hatchling. So this provides us with new information on the ontogenetics of Rhynchosaurs. There's only about 3 juvenile Rhynchosaur skulls preserved, and even then we didn't actually get a good look at the teeth usually. So we find that, we hypothesize that the ontogenetics of dentition in Rhynchosaurs likely began with the first year being like baby teeth in mammals. The teeth are acrodont, they are quickly either lost or worn away from use. And then the first set of adult dentition comes in, and with continuing years of growth, the tooth rows actually increase, and eventually we get the 4 to 5 tooth row that we see in the adult Bentonic skull. In addition to the new information about ontogenetics, we also see more about the evolution of Rhynchosaur dentition. The study by Langer in 2000 began to describe a lot of the paedomorphic shifts within Rhynchosaur evolution based on the dentition. And with the addition of our juvenile, we find that the what we assume to be Bentonics or Phodonyx juvenile actually has very similar dentition to a more advanced Rhynchosaur, Hyperdaptylon sanjuanensis. So that shakes up a lot of things. With the crazy questions of ontogenetics and evolution out of the way, we also come to the question of diet. The fact that the juvenile had different dentition than the adult could mean that the juvenile rhynchosaur experienced a dietary shift, much like modern reptiles can do sometimes. So we looked at a lot of different reptile fossils of similar sizes, and we found that the Capes bentoni, a procolophonid, had very similar dentition in the sense that it was round, pointed. And in procolophonids are herbivores and are possibly insectivores as well. Either way, they fed on a tough diet. So we are assuming in this case that the juvenile most likely fed on a tough diet, although it is unclear whether or not that diet was tough vegetation like the adults, or if we included other things like arthropods in there as well. Being able to CT scan rhynchosaurs for the first time also gives us a new opportunity to revisit tooth replacement in rhynchosaurs. As you can see, the adult exhibits the expected characteristics of rhynchosaur dentition. The tooth is worn down along with the bone, and it forms two shearing planes within the jaw. And we also see multiple rows of teeth, unlike that we see in the juvenile. Some oddities also exist in the adult as well. We find that there are no signs of replacement teeth in the maxilla, likely due to space restrictions within the maxilla itself, suggesting that tooth replacement only occurred in the posterior-most region, and it was very much more like a conveyor belt style tooth replacement. In the dentary, we see the typical rhynchosaur tooth replacement where there are new pulp cavities forming below the existing teeth, and they will likely slowly shift upwards and develop enamel caps and such, although it is much more disorganized in comparison to the more advanced Hyperdapodon. In conclusion, this juvenile is one of the most complete juveniles ever found. Evidence for major ontogenetic changes within the dentition, and evidence for paedomorphic shift in rhynchosaur dentary dentition as well. The morphological differences also suggest that there is a possibility of a dietary shift between juvenile and the adult. This gives us a new opportunity to also revisit rhynchosaur tooth replacement mechanisms in much earlier rhynchosaurs. In terms of further studies that we're currently undergoing, we're looking at reconstructing and doing some biomechanical analysis on the juvenile skull. And we've also received several new specimens of rhynchosaur jaws from the Otter Sandstone, also found by Rob Coram. I'd like to thank everyone who's helped me throughout this entire process, and thank you for listening.
Great, thank you so much, Perth. That was such a great talk. I had no idea that the rhynchosaurs wore down their jaw bone in addition to their teeth. Ow, let's just say ow. Well, thank you. If you have any questions about this great talk or any of our other amazing speakers for PalAS 2021 Mesozoic Life, you can go into the Slack channel and ask any questions that you want for the speakers, but also you can save your questions for the very end. In about 12 to 13 minutes, we're going to be doing an open question answer where I'll be monitoring the chat as well in order to kind of ask the speakers your various questions. So let's go on to our final talk of the day. It's Natalia Jagelska.
No worries.
Here Be Dragons, the perplexing patterns in the distribution, growth, and evolution of non-pterodactyloid pterosaurs. Thank you. Take it away.
Okay, I'll be sharing my screen. Da-da-da. Can you see? There's no problem. Yeah, okay, thumbs up. Okay, hi guys. And Victoria nicely introduced I'll be describing non-pterodactyl pterosaurs and what happened to them. But non-pterodactyl pterosaurs sounds like a lot. It's a lot. I struggle saying it. What does that actually mean? Well, with pterosaurs, we split them into two groups, non-pterodactyloids and pterodactyloids. We used to think they're separate and unrelated, but now we know they're related by modular evolution, but they still are very discrete. Non-pterodactyloids appeared in late Triassic and thrived till and start of Cretaceous, while pterodactyloids appeared in around Middle Jurassic and diversified in Cretaceous and were dominant creatures in Cretaceous before they went extinct with all dinosaurs. Non-pterodactyloids were the first vertebrates to achieve flight. They're quite small. I mean, they were still big. They were albatross size with wingspan around 2 meters and those long, elaborate tail veins, making them a very dragon-like. They were also very probably poor at terrestrial commotion, unlike pterodactyloids, which were much, much larger, with wingspans of almost 10 meters, the largest flying vertebrates, and probably better capable of moving on land. And probably when we talk about pterosaurs, you think about pterodactyloids. They are the pteranodon, the Quetzalcoatlus, and they're much more diverse and quite more interesting. Even when you look at the disparity results, pterodactyloids, which are in the blue, are much, much more diverse than small non-pterodactyloids. Still, non-pterodactyloids thrived for Triassic and Jurassic and were very successful. So what made them very successful in the first place, but they ultimately made them extinct by start of Cretaceous? So non-pterodactyloids appeared in Triassic. Their remains have been found in landlocked deserts, terrestrial steppes, dense forests, open seas, and lagoons. The basal forms disappeared in early Jurassic, and we had a new body plan of them appearing in Middle Jurassic, which spread globally. They had cosmopolitan distribution without relatively minor changes in Buckland, with no evidence of regional endemism, which makes sense. Back then in Triassic and Jurassic, we had one big continent and they're flying animals. You know, a river or sea will not be a big thing for them compared to a sauropods or other terrestrial animal. And we also can see the low disparity on display just here. We have these Capognathids from early, middle, and late Jurassic, and they look almost identical. If you plot them on phylogeny, they might form a big polytomy depending how you plot them, and they confirm different habitats. Dryognathus is marine and early Jurassic from Germany. Gigantognattus is terrestrial from China, and Scyphognathus is lagoonal, again from Germany. So very different environments, very different time periods, look almost exactly the same, no change. Big problem studying pterosaurs is that we don't know much about them, and nice fossils like the ones I showed previously come exclusively from lagerstatic conditions. And we know that they're global distributed, but most information about them, of non-pterodactyl pterosaurs, of pterosaurs overall, is just bits of bone which usually have no taxonomic value, disarticulated, and usually go under the radar. Also, presently they show us more about diversification and evolution, but sadly we don't have good access for them, or they're not that informative. We only can access lagerstatic condition specimens, which come with their own biases and don't show the full picture. In this talk, I'll be talking about two things that I sort of observed by looking at disarticulated material and completed material from Jurassic, and I suggest two processes that affected non-pterodactyloid and pterodactyloid evolution. For one, we had some kind of diversification or divergence in early to middle Jurassic, and some minor extinction and diversification of pterodactyloids at the start of Cretaceous. And it's not thing exclusive to pterosaurs. When you look at diversification patterns of dinosaurs, mammals, crocodiles, marine reptiles, everything on land and in sea, they follow a similar pattern. They develop in Triassic, they have the basal body plans. When you get to the middle Jurassic, the basal body plans disappear, and when we start developing new body plans, which became prominent in Cretaceous. And that appears to stegosaurids, iguanodontids, sauropods, theropods, and birds. And it's a similar thing we also observe in pterosaurs. So what did happen in Middle Jurassic to cause this enormous shift? Well, the problem is we don't know much about Middle Jurassic. We know very, very little about terrestrial Middle Jurassic deposits because they don't preserve that well. But I have a big solution, and I ask you for not tweeting because I'm showing a thing that's currently being reviewed in Current Biology. Fingers crossed it will get through the peer review. And it's a new Scottish pterosaur. And if you know anything about Scotland, it's amazing for Middle Jurassic deposits. Actually, it's one of the best places in the world for Middle Jurassic because we have entire stretch uninterrupted in Hebrides from that formation. We also have coeval formations from Oxfordshire region, and overall, Britain is great for studying pterodactyloids because we have record from lower to late Jurassic, almost uninterrupted, all yielding very nice fossils. So Middle Jurassic in UK is represented by two main fossil formations. One is Tatum limestone formation in Oxfordshire, which are shallow bay with terrestrial influences, and the Lealshire formation, which is a marginal marine deposit with Perlux suberyl exposure. allowing us insight for both terrestrial and marine environments. And Lield Formation is already exciting because it's one of the best track sites from Middle Jurassic, showing first evidence of Stegosaurus. So we know it's something that will displace this big diversification event as it is. And this is the wonderful specimen we recovered. It's looking like it's coming from some kind of Lagastata crater formation thing. Like it's from that formation, from the Alshire formation, we don't have any vertebrates apart from footprints. So this is an anomaly, and it's presented wonderfully for a pterosaur. It's in 3 dimensions, it's 70% complete. Only thing missing is the hind limbs and bits of wing phalanges. But everything is exactly, it's wonderful. And I've been working on describing it recently, and it fills this essential niche from Middle Jurassic. It's also one of the largest non-predator Jurassic pterosaurs we have on the record. Here's the largest one of the largest well-preserved non-predator on record from late Jurassic formation in South Volcano, which is the best formation for non-pterodactyloids. And here's where our specimen plots. It's way ahead everything we know so far, so it's a big boy. And our big boy is still growing. When we did histology of its wing phalange, it turns out it's a juvenile, at least falls within that range, which doesn't make any sense because osteologically it looks like a mature individual comparing to all sister clades we recovered. But its cortex is filled with fibular bone and just 2 lines of arrested growth, which, if you know anything about histology, means it might be a baby, which is crazy. That means it's still growing. And it doesn't make any sense because when you look at related animals from Jurassic of the same species, of the sister species, but even when they reach smaller, like 1 meter size, they already have a lot of aberration bone, which suggests they slowed growing. So we might be seeing some kind of anomaly in which some pterosaurs have very madly big histovariability. Our specimen is very unique. To see if our specimen is very unique, I look at other Baffinian specimens from other British Baffinian formations, which are not as well complete. It's just disarticulated bones. Woodland's bones are big, but they're just single bones. They don't have any taxonomic values. They still show us they're bigger than anything we expect. And when you try to extrapolate what kind of wingspans they had, we reach something around 4 to 3 meters, which is well below above the 2-meter wingspan, which we predicted previously, showing that they may be a big diversity of pterosaurs that is simply going missing because we're not looking at fossils coming from Lagastata formations. And there's a lot of this articulated material, which was a broader spectrum of animals, and that non-pterodactyls were much bigger than we anticipated. This articulated material from Baffinian formations from England also shows that those pterodactyloids and non-pterodactyloids started evolving in Middle Jurassic, and they remained exactly the same till late Jurassic. In the Baffinian formation, we have examples of scapognathids, aeroplonincoids, monophenestratans like Lepidopteroids and pterodactyloids, which only appear in Kimmeridgian Saltman formation, which suggests they remained exactly the same throughout this long period of time and live in the same faunal assemblages without changing that much, which is exciting. And also definitely shows us that they must have evolved before Baffinian and remained static up until Tiffanian or Kimmeridgian, which means something happened before Baffinian period. And what might have happened before Baffinian that might have driven this kind of diversification? I'm proposing some kind of solutions, but I still have to do some plotting to see how the fossil record collaborates with them. For one, Middle Jurassic was an interval of enhanced productivity driven by a shift to a more humid climate, enhanced by continental weathering and nutrient flux created by changes in ocean circulation. We had 2 big events happening. One was a Pliocene-Toarcian extinction event, which was driven by Toarcian anoxic event, which was associated with chiromagmatism and cooling period. We also had a thing called Middle Jurassic Revolution, which was a diversification of coccolithoporids and a major shift in ocean chemistry along Alien abrasion boundary, which might have both driven sort of kind of knock-on effect on marine and terrestrial environments, pushing them to diversify and try new body plans, which is also supported by global organization of oceanic plates. We have the central Atlantic opening and formation of Pacific plate, allowing creatures to have new niche-filling models of adaptive radiation. So, okay, they diversified in Middle Jurassic and thrived in the humid lagoonal environments with a lot of coccolithoporids. But why did they disappear in in early Cretaceous at the Jurassic Cretaceous boundary? What happened for this very, very successful group of non-pterodactyloids to disappear and be displaced by pterodactyloids? Well, in early Cretaceous, we have the biosalicyfication crisis, which means we had to reduce production of carbonates and more or less lethal environments. So if you don't have less reefs and coast, clades associated with those environments, like non-pterodactyl pterosaurs, are heavily affected. Also, we have disappearance of reef environments, which and more diverse climate, which allows more diverse animals that are better terrestrial capable and not reliant on marine environments to evolve, making pterodactyloids ripe to fill those niches and outcropping on non-pterodactyloids. Okay, to wrap up, seeing that I'm running out of time, the new fossil tells us that Middle Jurassic was a principal period for diversification of pterosaurs and other animals that includes dinosaurs, birds, and crocodile and just a plethora of animals. That they had static Buckland from early to late Jurassic, and maybe that kind of successful journalist thing that driven them from Jurassic was hit hard by environmental changes at Jurassic Cretaceous boundary and made them be ripe to be outplaced by their cousins, the pterodactyloids. It also shows amazing historical variation, showing that pterosaurs grew large fast, and they are very, very diverse when coming to the growth patterns, making them very, very unique animals that require more study. Okay, big thanks to everybody who helps me with the research. And if you want to follow me on Twitter for more information, I upload a lot of art. So thanks, Steve, thanks, Greg, and Mike, and everyone. And I hope you enjoyed the talk. And let's go to the Q&A. Thank you.
Thank you so much, Natalia. That is really interesting. What an amazing find in Scotland. Wow, I can't wait to see that paper when it comes out.
Hopefully January.
And just to remind everyone that you can go and look into the Slack channel of O2 Mesozoic Life to take a look at some of the questions that other people have asked our previous speakers, as well as any of their comments. And in fact, now we are on the group question and answer period. So if you have questions, please hop onto the talks. And if I can ask all of our speakers to go ahead and turn on your cameras, if you can, and let's look at some questions. I know you guys have been doing a great job of following the questions. I've been doing the best I can while trying to pay attention to everything all of you guys have been talking about in your talks, as well as taking some quick, looking to see what's going on. And I think we'll start with actually a question for Natalia, since your talk was the most recent, and also you haven't had a chance to hop on to answer any questions yet. So there is a question for you. Always my first thought when looking at diversity changes of Lagerstätte restricted taxa, how do the species numbers compare to area of rock exposed for the time? Lagerstätte rock area. And a comment, great talk, by the way.
Thank you. And yes, just looking at diversity estimates is hellish, because we are looking at something that when we see it preserved, that's just a fluke of nature that happened. That doesn't represent anything that's actually happening. So if we look at the Lagerstätte or even other tertiary environments outside of Lagerstätte conditions, we are not looking at the full picture as it is. Many people try to calibrate for Lagerstätte conditions on the circular material, but it's very, very hard because we are looking at freaks of preservation. And the problem with estimating diversity from in this circular material is that we have no taxic information on what kind of creatures there might be. It's just a pterosaur was there. I have no idea if it's a pterodactyloid, non-pterodactyloid, anything. It's just a wing phalange that looks pterosaur-like, but that's about it. That's not a good indicator of what kind of species thrived, how they were popular, were they in the optimal environment. So there were a lot of papers trying to counter the biases in pterosaurs, because pterosaurs have this ultimate clutch. They're very good at flying, but they're very bad at preserving because their bones are hollow and very easy to squash. And that might be also why we only find the overrepresented in Lagerstätte, just because in terrestrial environments, in which they might be very, very different, they just don't preserve as well or at all. And if they preserve, it's very uninformative on taxonomic level. I hope that answers your question.
Great. And just one follow-up question for that is, do you have a rough estimate of how big this new specimen could be?
Yes, we looked at Ramproncons and Orgnaphids, and all specimens well preserved, but sadly, we only have 2 wing phalanges and they disappear later. It's like, oh, we cannot do anything. It's so frustrating. But we are getting around over 2.5 meters as a minimum. And of course, it's still growing, so I have an idea how big it was. By looking at other specimens in Marfonian, we should be looking at around 2.5 to 3 meters, which, I mean, when looking at pterodactyloids, they have like 12 meters. So like that's a tiny, tiny rascal. But when comparing to things like frigates, that's still a big pterosaur, especially for that period, and just shows that maybe Big pterosaurs were thriving in terrestrial environments because they're in terrestrial environments, they don't preserve well, and we only find them in marine environments in the certificated states. So that means they're probably much, much bigger, and the things we are seeing with the smaller animals is just a preservation bias. They also grew rudely. The histology of pterosaurs doesn't make any sense. It's completely random. The sizes don't collate with histology. I have no idea what's going on. We need to do more studies, as people say. Somebody has to go to Oxford and just slice all those bones and see for the histology, how it correlates to the size.
Fantastic. Thank you so much. So with one of the next questions, forgive me, dog barking. So this goes to Mike Benton. What would the advantages of oviparity rather than viviparity?
Yeah, so people discuss why would it be advantageous to lay eggs, because if you carry your young in vivipary live birth, you protect them and you release them and help to feed them. I guess egg laying has many advantages, though, for our selected taxa, meaning they focus on high reproductive rate. You can produce a lot more young because you lay the eggs when they're smaller than a fully developed offspring, for example. You can have multiple clutches in a year, so certain birds may lay several times, which otherwise you probably couldn't do. There is a risk, of course, that you then typically abandon the young, but I think it's that sort of energy balance thing, and vivipary is the case-selected strategy, usually, maybe associated with poor climates as well. And laying eggs is an R-selected, maximized reproduction rate kind of strategy.
Thank you so much, Mike. Sorry. Now, I see Perth, you have got quite a long question or series of questions, and you have kindly added on to them. But so let me try and find that series of questions again. So one of the interesting things that has come up is, are you looking at the histology of the teeth? And I know you kind of mentioned that, but if you want to kind of elaborate anything about the teeth or the tissues with the jaws, that would be fantastic.
Yes, so it's a bit unfortunate, but at the resolution that we're looking at, the juvenile specimen and the adult, it's just unclear for us to look at any histology or anything like that. One of the big challenges of looking segmenting the specimen itself was also the fact that the density of the sediment surrounding it was very similar to the bone itself. So it's a bit of a challenge, and we have considered looking at the micro CT as an option, but that would require us to sort of destroy parts of the specimen to actually get it to fit into the micro CT itself.
Great.
So if I can cut in on that, because it was my question, I very much appreciate. Well, I very much appreciate the answer, but I also appreciate the difficulties. But since your talk gave so many insights, I think, on rhynchosaur dentition in general, and I don't think this has ever been done, I think, and it, you probably don't have time for this, but for someone at some point to pick up to look at just histology. So making sections of isolated, because I know these tooth plates are out there of different taxa. They can also be, there's abundant remains from South Africa. I think it's something interesting, because I think this is all quite unique, at least for stem archosauromorphs. Stem archosaurs or archosauromorphs. So it's very interesting stuff.
Yeah, it definitely is. Not a lot has been done with rhynchosaurs, especially regarding rhynchosaurs and CT scanning and things like that. So there's a lot to be done.
No, thank you so much, Stefan, for following up with that, because and you have done some amazing things with your CT scanning. I mean, your CT scans, Stefan, were absolutely beautiful that you were showing from the whales material, which is in such an astonishing quality, especially in the three-dimensional shape. Have you had any challenges with that? I mean, I know that whales, dinosaurs, and other archosaurs are not terribly common, but you have definitely done some amazing work on them.
Yeah, actually the scan was, I'm very happy with how it ended up, but it was a lot of work. It was really one of the more difficult scans I've worked on. And this is mainly because everything in this crocodilomorph is so pneumatized. The skull is very pneumatized. The vertebrae as well are very, very hollow, actually. So it was very, very difficult to actually trace the bones through the scan. So it took a lot longer than I anticipated it to take, but quite happy with the results. And it's actually something, yeah, I'd be interested to look more into, is this pneumaticity and all of that, and also whether it might also be a preservational thing, because I know, you know, multiple people work on the Fisher film material, and CT scans have been made as well, and this material is also notorious, I think a little bit notorious to work on also from a histological perspective, that maybe some of the bone is also dissolved. So this is something, yeah, I'm somewhat curious to whether the difficulties with the scan were attributable to the taxon or the preservation. So yeah, we'll see maybe going forward what this is all about.
Oh, fantastic. No, you definitely have some, all of you have incredible specimens that you showed. I was going to ask, Ood, your amazing pterosaur, you have, you've shown some great feather structures and feathers with melanosomes in them. Have you also looked at the actual crest itself as to if there's any organics preserved in that crest?
Yeah, so that's something that we plan to do. So we have sampled the skin. So there is melanosomes in all the elongated fibers. So yeah, the next step now is to look at variation in geometry in the skin, in the soft tissue crest. But yeah, we haven't done that in detail. Haven't looked in detail yet.
Oh, that's fantastic. I mean, it really, I'm really excited to see that one, especially with the soft tissue preservation that you're getting. And I'm curious about what chemistry you guys are going to end up doing as well on it. And I'm sure that's something that we can follow up on at some point. I do want to follow up also with Thomas. I am so sorry that I had to cut you off because your talk was so fascinating. And I think it's something that all paleontologists need to kind of learn more about and be more aware of, especially when naming new dinosaurs or other animals or plants even as well. Do you have any follow-up comments that you'd like to kind of follow up with?
I mean, first of all, you don't need to apologize. That was my fault for overrunning. And yeah, secondly, I think, I mean, if we're talking about what actually we can practically do about this, then the first thing to say is that, you know, even when it's not legally mandated, collaborate with people from the country in which the fossil has come from which the fossil has come. So countries like Brazil, which have it as a sort of legally required condition of publication, really, that you collaborate with and include as an author somebody from that country, they really buck the trend for the inclusion of those authors as first authors, right? So it sort of leads to a more active participation. But also there's an element of engaging proactively with people who, if, for example, you want to name a species in a language that you are not fluent with and none of your authors are fluent with, as could well be the case, even if somebody is from that country, then really engaging with the community and asking native speakers to have an input on the name is crucial, because there are instances of translation errors and places where you end up with names that are not particularly appropriate. So a good example of good practice is Ugranaluk cookpicensis, which is I think a hadrosaurid. It's a dinosaur that was found in Alaska fairly recently. And with that, the scientists consulted with the Inupiat-speaking local community and actively acknowledged them in the paper. And they came up with a name together that is respectful to local beliefs about ancestors and past inhabitants of a land. Whereas some of the Mongolian dinosaurs that have been described recently, so one of the most recent is Kurukula, which is named after a Buddhist deity, but it's the name of the deity is one word, Kurukula, and it's been split into the genus Kuru and the species Kula, which means that if we ever find another species of that genus, then it's sort of half the name ends up not really honoring anything. And in the description, they even say that it's not to be confused with Kuru, which is the name of a cannibalism-borne prion disease, right? So the fact that they need to sort of make this choice to split the name and then justify that it's not about something completely horrific and unrelated is really quite sort of unhelpful. And there's also dinosaurs like Nguevu Ndloko, which is a dinosaur named in Mosa, and that's a it's supposed to mean grey head, but the Nguevu, in that language, adjectives always have to follow nouns, but it's the other way around in the name, and they haven't included the root which would match the adjective to the noun case. So it's basically just not understanding the grammar of the language, looking in a dictionary to find out what the root is of that word, you know, and taking that as the whole word and not really understanding how it's constructed. And in the ICZN at the moment, Article 31.2 does say that you have to make sure that if it's from a Latin word, the grammar is consistent with the grammatical gender. But to restrict this only to Latin and Latinized words seems to be, you know, completely non-inclusive. So we are discussing among our group proposals that could be submitted to the ICZN to either clarify some of the language or to put some of the recommendations into articles. We're trying to see what is practical, given that we don't want to completely upend taxonomy. That would cause far more problems than it would solve. So we have to some extent, deal with the names that are in existence at the moment, and just try and have best practice going forward.
That's fantastic. No, wow, we will all, I'm sure, keep an eye out for any changes, because that impacts all of us, irrelevant of what area you're researching. Any name changes will definitely affect all of us, or how to approach and how we really should think about it. Well, thank you to all of our speakers. And we are going to be leaving this chat open. So if people have further questions for our speakers, please take a look. There are a couple of questions that I unfortunately did not come up to towards Mike. But if you want to continue these chats during our coffee break, which we have now until 11 o'clock, and then that follows on to our next series of sessions. So thank you, everybody, for your great talks. And I look forward to reading more about the questions and answers in the chat. So again, I'm sorry we couldn't all meet in person here in Manchester, but the PalAS committee has done an amazing job putting everything online on such short notice. Thank you, everyone, for doing all of this for us. Have a good morning and have a good afternoon. Bye.